So keep in mind the central dogma of biology, right? That DNA self-replicates and then it is transcribed to mRNA. So we'll talk a bit about this process of transcription today. And of course, there's a whole host of enzymes that are responsible for both DNA replication and transcription, namely polymerase enzymes. And then from messenger RNA, we then get translation so that the mRNA sequence is read in three codon or three nucleotide segments known as codons, and that's going to allow us to form a protein, which we know is just a polymer of amino acids. So let's talk a bit about the structure of RNA first. So RNA is less stable than DNA. And part of this is because RNA is not really meant to stick around for a long time, or its half-life is much, much shorter than DNA, where DNA is the permanent storage of information, and therefore it must be much more stable. RNA is more a temporary storage of information. So here I have the phosphodiester sort of backbone of an RNA segment, where we would have a phosphate at the 5' end, and then if this were the end of the chain, there would be a free alcohol at the 3' end, but I'm showing that it's connecting through another phosphodiester bond to another nucleotide at the 3' end. And we talked briefly about the fundamental difference between DNA and RNA last time, where we said that DNA, right, has deoxyribose pentose rings, but in RNA you have this C2' hydroxyl, or it's actually a ribose ring, right? It's derived from a 5-carbon sugar. So this hydroxyl at carbon 2 is going to confer some instability for RNA. And the primary example of that is known as alkaline hydrolysis. Alkaline is just a fancy term for basic solution. And hydrolysis means that water is going to serve as a nucleophile and break some bond or kick out a leaving group. Now, in basic solution, what can happen is hydroxide can deprotonate this proton to some equilibrium extent. So I'm going to copy this structure and draw the anion that forms there. So now we've generated essentially a much better nucleophile. And that nucleophile oxygen is 1, 2, 3, 4, 5 atoms away from the adjacent phosphoryl group of the phosphodiester bond. Right? This phosphodiester is keeping the RNA polymer intact, just like it did for DNA. And so what will happen next is the oxygen will attack that phosphorus. We know that 5-membered rings, right, are pretty stable transition states. And perhaps the pi bond would break. I'm showing this an abbreviated mechanism. The pi bond would reform later, and then this leaving group would leave. Okay, so that's really 2 steps, but I really just want you to see the product here. Just like carbonyl chemistry, that could go through addition elimination. And then what we're going to have are 2 separate products, right? This arrow here shows that phosphodiester bond breaking. So you'll have the 5' end, where now we've got the cyclic structure. So that 3' alcohol is still attached to the phosphorus. And there are still 5 bonds between phosphorus and oxygen, but now the secondary alcohol is also attached. So that makes a new phosphodiester. And we simply cleave off the 3' end. It's a separate sugar. And that would eventually get protonated to make this neutral alcohol. So there'd be some proton transfer to get here. And that would be connected to the rest of the chain at the 3' end, but this could continue, right, for every single RNA nucleotide in the polymer. So this hydrolysis effectively cleaves the phosphodester backbone. And RNA, through this hydrolysis mechanism, is essentially self-degrading. So if DNA did that, we would be in trouble, right? That's why DNA lacks this C2' hydroxyl and is much more stable. So this, of course, has some evolutionary benefit, right? And I mentioned it briefly before. DNA replication and the process of making DNA has a much higher fidelity, which means that it makes fewer mistakes. Than the processes of RNA metabolism, right? Transcription and translation. So DNA replication has many sort of double check mechanisms, such as mismatch repair and other proofreading techniques. Whereas we'll find out when we talk about transcription today, that transcription is not nearly as high in fidelity. And the reason for this is because organisms that have mistakes in their DNA replication, right, are going to go on to proliferate those mistakes for every daughter DNA strand that is replicated from that parent that was somehow incorrectly made. So a mistake in DNA is much more consequential for a cell. And therefore, these mechanisms of double checking the DNA polymerase machinery is going to be much more predominant for DNA metabolism. If you have some mRNA strand that is incorrectly made, because RNA polymerase will make mistakes, it's not that big of a deal because each mRNA strand is just going to go and produce a protein. So then you'll have one faulty protein with the incorrect amino acid somewhere in the sequence. And then that protein can just be degraded. And the protein does not go on to actually serve as the template for any replication of other proteins, right? But DNA is that essential source of information that must have a high fidelity in its replication. So we talked about how DNA is primarily double-stranded, right, in eukaryotes. And it turns out that RNA much more commonly is single-stranded. There is double-stranded RNA, but we're not really going to see it much in this course. So that's a big fundamental difference between the secondary and tertiary structures of these nucleic acids. Right? DNA forms a double helix, and single-stranded RNA does not. So we won't talk much about a folding process because we simply have a single single nucleotide strand or a single nucleic acid, in most cases for messenger RNA, tRNA, etc. Okay, so that's not to say that RNA doesn't have unique three-dimensional structures, right? So one of those common three-dimensional structures that we're going to see a little bit in transcription is the following, where I've shown the linear sequence of some nucleotides here, and you'll notice that guanine is going to undergo Watson-Crick base pairing with cytosine, and adenosine will hydrogen bond correctly with uracil, and this self-complementary sequence keeps going. Cytosine with guanine, once again, cytosine with guanine, uracil with adenosine, and finally guanine with cytosine. And that's where the complementary component ends, right? Because now A and C don't match up to make the correct hydrogen bonding. So when you have this self-complementary sequence, what you'll find is that those RNA strands often form hairpin three-dimensional structures. Or we get this process of self-hydrogen bonding. So up here in the non-hydrogen bonded portion, we'll have A, C, U, C. And then towards the 5' end in the hydrogen bonding segment, we're going to have guanine, adenosine, two cytosines, uracil, and guanine. And I'll put the hydrogen bonds here in green. And you could be asked to count the number of these hydrogen bonds based on knowing the Watson-Crick base pairing rules, right? So every G and C will make 3 hydrogen bonds. Every A and U will make 2. And then towards the 3' end, right, we have C, U, G, G, A, C. And the covalent bonds there are shown in blue. So this is sort of our hairpin structure. And we often see this hairpin formation occur during what's known as intrinsic termination of transcription, or this is when the RNA polymerase enzyme is told to stop transcribing DNA into mRNA. So when you get this self-complementary sequence, the RNA begins to hydrogen bond with itself. And form a hairpin, and that causes it to dissociate from DNA, since the hydrogen bonds between RNA and DNA, or the hybrid, are broken. So you'll often find these self-complementary sequences towards the 3' end of a growing mRNA strand, at the point at which that gene has been finished in terms of it's appropriate transcription. It's sort of a stop signal. Okay, so if you have a hairpin, of course you have to have that Watson-Crick base pairing, where G and C make 3 hydrogen bonds, A and U make 2. But in general, right, because we don't have single-stranded RNA, with the exception of a hairpin, there are no Chargaff's rules that can be applied, right? Recall that for double-stranded DNA, we know that the percentage of A was equal to T, and the percentage of G was equal to C. And if you just know one of these, you can find all 4 because all of them sum up to 100%. But for single-stranded RNA, we can really have any combination of A, U, G, and C. So Chargaff's rules do not apply unless it makes some sort of hairpin structure. And you'd have to be told that. So, for example, you could be asked a multiple choice question. There are multiple in the test bank like this where you're asked to select any of the following combinations of nucleotides that could be the constituents of a single-stranded RNA structure. And as long as they sum to 100% and you've got the appropriate nucleotides, cases A and B, those are fine. But of course, C is incorrect because this has thymine, which is present in DNA instead of RNA. So watch out for questions like that. A lot of people get caught up thinking that it's DNA and therefore the base pairing rules will tell you the percentages. And here in single-stranded RNA, they don't.